All right, so thanks again, everyone, for coming out tonight. We're really looking forward to tonight's speaker, who is Dr. Alan Weakley, and his talk is Taxonomy, Floras, and Plant Identification Tools for Biodiversity Conservation in the 2020s. Alan is the Adjunct Associate Professor, the Director of the University of North Carolina Chapel Hill Herbarium, and that's his official title, but I like to think that many of us involved at field botany think of him informally as the chief regional botanist of the Southeast and the guru of Southeast regional flora. He completed his PhD at Duke with a thesis, Change Over Time in Our Understanding of the Flora of the Southeastern United States, Implications for Plant Systematics, Bioinformatics, and Conservation. As you can see from this slide, he took an early interest in botany, and he announced his interest by blowing on the trumpet of Datura at the age of, looks like about 7. So Dr. Weakley has been very involved in citizen science and public outreach and has many innovative approaches of great value to the field botanist and regional botanists. And that includes these new set of regional floras, which are complete with dichotomous keys, synonymy, and loads of references. And I just thought I'd mention a New England connection that he's been assisted in the work in part by 2 former Massachusetts and New England botanists who've been working for years with him, Richard LeBlond and Bruce Sorey. And Bruce Sorey, of course, was the first state botanist, and many of the records that I review every day for my work are full of the valuable observations of those 2 highly skilled botanists. So Dr. Weakley's lucky to have them on his side down there. So we're looking through a few of these slides of the regional floras. These are the titles, and we're gratified to see excellent photos from Professor Bellemare, our former president now, who went from his visits to the Southeast, maybe on that very early trip he took, led by Alan Weakley. So these regional floras are all publicly available, and you can download PDFs of them and look through the keys. And they are often very helpful, even up here in New England. One of the ones we didn't show was a coastal plain flora that really comes up into New Jersey and then very close to Long Island and on up to our Cape Cod and the like. So, but of course, many plants have a broad range, and so you'll find the addition of an extra key very useful at times for particular species. And Dr. Weakley's a prolific photographer and doesn't only focus on rare species. Here's one of my favorites that he took and posts with oak leaves that he found on the forest floor. Didn't do any rearranging to this at all, but just snapped the image. And there's, I think, about 20 species of oak and a few other things in here. It's just remarkable, showing the diversity of tree flora down there in the Southeast. And I want to just add one little bit before we have him speak, and that was from a post about the same time as this picture, where he had some eloquent words about sort of the project in general and about what's going on in the Southeast, and it was very eloquently done. So I'll just include that here before his talk. Dr. Weakley said, "But I think the largest factor is that we are overcoming a collective failure of imagination." Imagination, you say? Isn't that the antithesis of science? I'm not talking about making stuff up. Science is always based on imagination, and certainly in biodiversity exploration, we see what we expect or are willing to allow ourselves to see. We can walk past the lobelia muttering, one of those coastal plain wetland lobelias, or we can look at it and examine it because we're someplace interesting and realize, wow, that's different. At the core of our collective failure of imagination is that we simply haven't appreciated the tremendous diversity of different habitats and the great age of those different settings in the highly variable and ancient, though ever changing as well, landscapes of the southeastern United States. Different rocks, different microclimates, different fire regimes, different hydrologies, all of those combinations, isolating lineages and spitting out separate evolutionary entities throughout the eras. So with that, Alan, I'd like to welcome you to New England Botanical Society.
Thanks, Bob, for that intro, that elegant intro. I'm a little bit embarrassed by it, I guess, but I'll strive to get over that and move on forward with the talk. So thanks for inviting me to speak with you all, to be with you all this evening, and I hope be able to keep it interesting and fun. So as Bob has already said, I've been at work for about 3 decades now on a new flora for the southeastern United States, broadly defined. And that area does include all of the unglaciated southeast out to what I call the dry line in Texas and Oklahoma. And in order to conveniently round off a few states, it includes some glaciated bits up your way too, in order to include all of New Jersey and all of Pennsylvania. So the southeastern plant diversity is rich, and I'm not going to go into a lot of detail in this talk about the sort of reasons for that, but the paragraphs that Bob quoted me on sort of talk about it some. And unfortunately, in the Southeast, we have a kind of a mismatch between high biodiversity and low conservation. And therefore, I feel like we are really pressed, and it is really urgent for us to do the best job we can to provide good tools for finding biodiversity, for identifying biodiversity, for mapping it, and then working to protect it, and all. So I've been really spending a lot of time and energy here towards the end of my career and trying to develop those kinds of tools. So I want to talk a little bit first about taxonomy and change in taxonomy, and then I'm going to talk about floras and tools and so forth. Taxonomic change through time. You know, we all kind of bemoan that, this old friend that we used to call by one name, its name has changed. Those damn taxonomists, they're at it again, etc. But this is the process. It's always been the process. In my dissertation, I did an analysis of the level of name changes and taxonomic changes through time in the Eastern North American flora, and actually the changes are less now than at many times in the past. But there are new paradigms and new definitions that affect this. There is also the matter of scientific fashion, and I'll talk briefly about that. And that gets into that perennial question, what is a species? So this is something we don't really think about very often, but I think it's an important thing to remind ourselves. And that is that most of the species of Eastern North America were named under a typological or morphological species concept. They were named in the 1700s and 1800s before evolution was thought to exist. All those species named by Linnaeus, he was naming God's special creation that had been placed on Earth for Adam and Eve and their descendants to enumerate. And so a typological definition, there are entities that exist as a limited number of universals or types recognizable by morphology. These types do not bear any relationship to each other. The universals or types are called species. Variation is considered as trifling and irrelevant phenomena. But the modern species concept that we operate under is more an evolutionary species concept based on descent and relationship. An evolutionary species is a lineage, an ancestral descendant sequence of populations evolving separately from others and with its own unitary evolutionary role and tendencies. There are various different phrasings of some of the details of that and different emphases on lineage or relationship or gene flow and so forth, but that's basically a modern species concept. And these different species concepts matter. They matter a great deal. So something named under one species concept may or may not actually be a good species under another one. And we are essentially in a process of reevaluating all of the species taxonomy done in the past. And one of the things I also studied in my dissertation was kind of scientific fashion. And I hope you won't take offense at me using the beloved Gleason and Cronquist 1991 as an example of this. But essentially the period from about 1968, the publication time in the southeast of Radford, Allison Bell, Manual of the Vascular Flora of the Carolinas, up until the early 1990s, and for instance, Cronquist's Flora, published in 1991, was really a period where lumping was fashionable. And it's sometimes said that this was a reaction to the excesses, so-called, of Small in the Southeast 1933 Flora, and Fernald in his the 8th edition of Gray's Manual. But a lot of the lumping was done with no study or very minimal study. It was often done just in floras. It was often done with nary an explanation or analysis, or perhaps a short sentence, and therefore no published account of why things were being lumped. So it was partly, I think, a burden of proof issue where people said, yeah, well, I don't think the previous authors proved that that was a species. But you can argue, I guess, on where the burden of proof does lie. They published it as species, it was accepted as a species, and then later it was just sort of swept under the rug with little real further analysis. As I already said, lots of lumping done with no or minimal study, no published account. So I'm going to talk about a couple of examples that I think are maybe illustrative about this. And the first is the viburnum nudum complex. And I think many of us are familiar with the idea that in Eastern North America, there are 2 taxa. Sometimes they're treated at species rank, Viburnum casinoides and Viburnum nudum, or sometimes as Viburnum nudum with 2 varieties, Var nudum and Var casinoides. And the Flora Novae Anglii has that varietal treatment. Cronquist 1991 has that varietal treatment, and so forth. 2 taxa. Controversial. And the distribution maps in the Biota of North America program looks like this, with Viburnum casinoides being generally more northern and coming down the Appalachian Mountains, Viburnum nudum being mainly more southern and especially coastal plain, and reaching up to southern New England. But if you actually look a little bit closely at these maps, you may notice some problematic things. So what's all this casinoides down here in the deepest south, where you might expect casinoides to be more of a Yankee thing? Beth Spriggs, working in the Michael Donohue lab at Yale, recently tackled this group and published a paper in 2019 entitled "Restriction Site Associated DNA Sequencing Reveals a Cryptic Viburnum Species on the North American Coastal Plain." This is in contrast to Cronquist's statement, 2 geographic varieties often treated as distinct species, but apparently overlapping in all characters. That's the kind of comment that basically led to a lot of lumping, or that was used as a rationale for a lot of lumping. Oh, we can't tell these 2 apart reliably. We can't sort the specimens in the herbarium with 100% confidence, and therefore we don't think there are actually 2 taxa. Beth used a variety of means, and what she determined in her study was that there were 3 entities. And this southern casinoides-like thing was more closely related to Cassinoides, but had been being lumped into Nudum. So there was a cross relationship of the two that and what the actual relationship was determined to be was contrary to the way the taxonomy had treated it. So if you look at the black dots, these are the mapping of herbarium specimens of Cassinoides in the upper right, and Nitidum in the middle in the red here, and then Nudum in the green on the lower right corner. So 3 taxa. And she writes, the view is widespread that plant species are often difficult to delimit due to hybridization, polyploidy, asexual reproduction, and phenotypic plasticity. At the outset, the Viburnum nudum complex appeared to be a classic case of such difficulties. However, it turned out to be surprisingly straightforward, and this offers hope that the confluence of molecular, morphological, geographical, and ecological data along the lines demonstrated here will prove to be the rule in plants rather than the exception. Our molecular analyses have clearly identified 3 distinct lineages, one of which, Viburnum nudum, is highly divergent from the other 2. The morphological differences among the 3 clades are minor, but Viburnum nudum can be distinguished with considerable confidence based on morphology alone. The more recently diverged Viburnum nitidum and Viburnum cassinoides are not so clearly separated based on morphological traits, but molecular evidence indicates that these 2 clades are isolated by more than geographic distance, and they appear to have maintained their separation through significant geographic movements during glacial cycles. Based on all of the available evidence, we hypothesize that these 3 lineages are evolving independently, and we therefore recognize them as separate species. So it turns out that a bird watcher, William McAtee, who published a monograph of Viburnum in 1956, privately published by the author in Chapel Hill, North Carolina, he was pretty much right way back in the 1950s. He had Viburnum nudum and then 3 varieties of Cassinoides, var cassinoides, which corresponds to Beth's cassinoides, and then the other 2 varieties, Nitidum, a coastal plain, and Harbisonii, some isolated mountain populations that correspond to her Viburnum nitidum. So this was based only, of course, on morphology. He distinguished that these things were good taxa. So I think a number of lessons could be drawn from this. One is that we should be humble about what we can or cannot tell based on morphology alone, but that when we do combine, as Beth indicated, a variety of types of evidence, we are likely to get to an answer that is closer to the actual underlying truth. My second example is with the blue Baptisias, the Baptisia australis complex. And this Baptisia australis, this one, Baptisia minor, out in the prairies in Kansas. And this one, Baptisia aberrans, the aberrant southern Baptisia, that occurs on glades in North Carolina, Tennessee, Kentucky, and Georgia. And Baptisia australis was named by Linnaeus in 1767 in the genus Sophora. Baptisia minor was named by Lehmann in 1827. Aberrans was first recognized as a variety by Mary Maxine Laracy in 1940, but then since then was largely and usually lumped variously and illogically into either australis or minor. And she wrote in 1940, Baptisia australis has for many years been the classical dumping ground for material collected in the Middle West. There can be little doubt as to the true nature of Baptisia australis because for no other species is there such a wealth of literature copiously illustrated. Early in the 19th century, it was introduced into European gardens, etc. So Cronquist in 1991, again, treated these at varietal rank sort of grudgingly. He didn't mention aberrans, which was in his range, but he treated australis and minor as varieties, contrary to Laracy's monograph in 1940. And he wrote, var australis is replaced west of the Mississippi River by var minor with smaller flowers, slightly larger flowers, smaller leaflets, slightly larger flowers, and more inflated and more evidently stipitate pods. A number of morphological differences, as well as the geography. So here is Baptisia minor out in Kansas and Oklahoma primarily. Here's in prairies. Here's Baptisia australis of river scour communities scattered through the broad Appalachians. Not all these dots are correct. And then here's Baptisia aberrans. Once again, some of these records are not correct, and some in Kentucky should be it. But with a more southern distribution in limestone glades and diabase barrens. My third example is the genus Marshallia, in which my lab has named a couple of new species. The first was Marshallia legrandii. This we initially described just based on morphology. It was later supported by some DNA studies, but it is a 2-site endemic, and a conservation philanthropist recently spent $12 million to acquire additional habitat for this species, a 7,500 acre addition to a Virginia Natural Heritage Preserve that will provide much additional habitat for this very rare species. So taxonomy matters. It can inspire conservation. And then later, Derek Poindexter, Wes Knapp, and I also determined that there were some needed taxonomic changes in Marshallia involving Marshallia grandiflora, which had been a name broadly applied to material from North Carolina and Tennessee north up to Pennsylvania. But it turned out that based on morphology and other factors, ecology and phenology and so forth, that Marshallia grandiflora is apparently extinct. It only, as far as we know, had 1 or 2 populations down in southwestern North Carolina. Here's Marshallia legrandii, and then what had mostly been previously called Marshallia grandiflora required a new name, Marshallia pulchra. So we have 3 rare species, one purportedly extinct, now where once we had 1 species. Recognizing these distinctions is important. So Beth Spriggs, in the paragraph I read, she talked about the need to basically bring all evidence to bear on these problems. Molecular evidence alone does not tell us what we need to know. Molecular evidence doesn't really inform us whether a lineage is independent and separate, and so forth. Morphological evidence alone maybe also isn't perfect. I think we really need to use all things, including geography. Tropvateria caroliniensis has been considered to be a single species with 1 variety in Eastern North America, 1 in Western North America, and 1 in Japan. Not really a credible thing, and molecular evidence is now suggesting that we have 3 species here in Eastern North America, 2 in Western North America, and 1 in Asia. Ecology can be an important piece of information to consider. This is the Buck Creek Serpentine Barren, and a recently named, Dave Buford at Harvard was one of the namers of this, Pacorus serpenticola, morphologically distinctive and isolated in this very unique habitat, and a habitat, serpentine barrens, that is known to generate endemism and isolation. Ecology, this slide from work done by Derek Poindexter in my lab on the Montani group of Carex, very different habitats of multiple taxa. Phenology, one of the clues for us on Marshallia legrandii is that it was in flower growing side by side almost 2 months later than Marshallia obovata, with which it was co-occurring. So there was phenological separation and therefore no opportunity for gene flow between these two population systems. Morphology. Morphology has been the mainstay and the go-to in the typological species concept. It still is a critical feature that tells us a lot of what we need to know about taxonomy, or provides us a lot of the evidence that we need to know. And of course, we can analyze that with statistical methods. And this again is looking at micromorphology and doing very careful analyses of the shape of paragenia, the vestiture on the paragenia and so forth, to tease out differences in the kind of cryptic Carex montani group. And genetics, DNA analyses, looking at phylogenies. So here we have Carex pennsylvanica and two things that had been previously lumped into pennsylvanica. They come out separately in genetics, they come out separately in morphology, and they also have different chromosome numbers, N equals 18, N equals 21, and N equals 17, as well as different habitats. So different geography, different habitats, different chromosome numbers, different genetic lineages from DNA analyses. So bringing together all of these multiple lines of evidence gives us a new synthesis of taxonomy that enables us to better understand the true diversity that is around us. So this brings us to a kind of unfortunate or complicated conclusion, and that is that species do not evolve for the convenience of human taxonomists. And it's a hard thing for us to fully accept that there might be two species which are that completely meet the criteria for being two separate species, but that are morphologically overlapping enough that our human brains and our means of measuring and looking at species without our DNA gun, may not enable us to reliably always distinguish them. So once two lineages separate, they are under no obligation to evolve characters to enable their distinction in the field or herbarium by human senses. Fortunately, they usually do develop such characters. Another little maxim, I think, is that geography and habitat are destiny. When things are separated into different habitats and different geographies, they will tend to diverge over time. So use all evidence and then make taxonomic judgments and write it down. Describe why you made the reasoning that you made based on what data you made, so that future workers can move forward rather than redoing something that you did and didn't actually record or publish. Okay, so I'm going to turn to a slightly different subject and start working towards talking about the tools. So the level of taxonomic change, the description of new species, is larger than most people think in Eastern North America. We are very actively still doing finding and describing new species, and they aren't all sedges. Some of them are rhododendrons and orchids and so forth. Something like 10% of the native flora of the southeastern United States, about 6,000 species total, about 600 of them have been named or are being named in the last 60 years, 70 years, 60. And many of those species that are being named are rare species. They're narrowly endemic species of specialized habitats. In the NatureServe lingo, G1, G2, or G3 species, the more imperiled end of the conservation spectrum. And therefore, these species warrant conservation attention. They warrant legal listing in many cases, and so forth. Perhaps more than 50% of all the G1, G3 species in Eastern North America have been named in the last 50 years. And this makes considerable sense if you think about it. Things like white oak were encountered early and described early, and so forth. And it is as we work into more specialized habitats, look more closely at the flora, that we're discovering a lot of these narrowly endemic species. That process of naming new species is not asymptoting towards zero either. In fact, we see the number of new species named actually increasing by decade. And so part of this is the new criteria, the new paradigm of what is a species. And part of it is new tools like DNA analysis. And part of it is simply access. When J.K. Small wrote his Flora of the Southeastern United States in 1933, there were large parts of the Southeast that had really had very little collecting done, and special habitats that had never been encountered by botanists and documented. So just in the last 2 years, I think there have been 4 species already named in 2022 in the Southeastern United States. But in the 2 years of calendar years of 2020 and 2021, all of these new species were named in the Southeastern United States. And as I said, there are some obscure grasses and genera like Dicanthelium and so forth, but there's tiarellas and rhododendrons and liatrices and fothergillas and so forth. So, and you can see the habitats and the approximate G ranks for these species, many of them rare and imperiled. So it behooves us to update floras, but floras have traditionally, floras are where the rubber meets the road. Floras are what makes taxonomy accessible to most users who are not going to be accessing 5,000 papers in miscellaneous journals, largely behind paywalls. So floras is where the rubber meets the road in terms of our knowledge of the plants in a region. And it's worth mentioning briefly that there are actual changes in the flora of a region as well. So for instance, Ficaria, which is becoming a terrible weed in the mid-Atlantic and parts of the Southeast. And I know that there have been alerts for it up in the Northeast and New England as well. Fernald in 1950 called it an old garden plant occasionally spreading to waste places or open woods and locally established. Cronquist in 1991 said casually escaped from cultivation in our range. That casual word used in floras tends to mean that there's not much of it. It very rarely escapes. It only slightly escapes or whatever. But now we have many situations across Eastern North America where Ficaria looks like this bottom left photo, dominating, completely smothering out other vegetation in a floodplain situation. So with increasing urgency for conservation, I think it merits us making extraordinary efforts to find biodiversity, to describe that biodiversity, and to conserve the biodiversity. And so that involves publicizing it with new global biodiversity hotspots, like this paper that Reed Noss and I and others wrote a number of years ago about the coastal plain, adding the North American coastal plain to the list of biodiversity hotspots of the world. And recognizing that the level of alteration and conversion of the natural landscape across Eastern North America is very high, and the ability to support biodiversity is steadily decreasing. And in fact, in a paper from colleagues at Duke, they showed that U.S. protected lands mismatch biodiversity priorities very substantially, particularly across the Southeast broadly and on the West Coast as well. So Type 1 and type 2 errors in conservation. The conservation of biodiversity is urgent. We have limited resources for land conservation and species conservation. We need to apply those resources as strategically as possible. We don't want to make either kind of error, a type 1 error where we falsely do not recognize a species as good that is, and therefore take no action to conserve it, nor do we want to make type 2 errors where we falsely do recognize a species as good that isn't and waste resources on its conservation. Neither of those are good outcomes for biodiversity and for our efforts to conserve it. And I just want to make a slightly political comment that conservation is conservative. The definition of conservative is tending or disposed to maintain existing views, conditions, or institutions. And nothing is less conservative than plowing ahead with the massive conversion of the Earth to a system that we do not know will be able to support us into the future. Okay, so I want to talk about tools. So plant blindness. We currently have a situation where arguably fewer and fewer people are really in touch with the natural landscape. There's less education in botany and natural history in general. There's less funding from federal sources like NSF for things deemed so 19th or 20th century as, quote, natural history. Universities have turned towards medical biology and the funding available there, and so forth, and all. So botany is not in as good a stead as maybe it once was. And Bob already showed you this photo, but I use it as an example of plant blindness. My parents were actually quite into plants and got me interested in plants at an early age. I think I was actually 5 at the time this was taken, but they probably should not have been allowing me to play as a trumpet, a terribly poisonous and hallucinogenic plant, even if it was grown in a garden. So Floras, plant guides, apps, websites. I think we need to synthesize information on biodiversity and make it more accessible to anybody and everybody. In the Southeast, we have had poor coverage of state floras. Many states have never had a flora, Alabama and Mississippi. Alabama is one of the most biodiversity rich states in the country, never had a state flora. Old floras have out-of-date taxonomy and information. If you're botanizing in Alabama until the recent decades, you used J.K. Small's flora, or you used a flora like Radford Allison Bell from the Carolinas that didn't really cover Alabama well. We write floras still in kind of 19th century formats. We write big tomes and books that we can't revise easily, and we publish them in these kind of old-fashioned formats. There are now new generations of users of botanical information who have different expectations. We need more people who could wear this, my favorite t-shirt. So floras are old, they're impractical to update. They get the information in them becomes skunky. And so what I've been trying to do over the last 20 or 30 years is reinvent the flora as a 21st century tool for biodiversity inventory, not your grandmother's flora. We need these tools to power community and citizen science, to power the natural heritage programs, the state parks and wildlife agencies, etc. So when we wrote the Flora of Virginia, it was the first flora for the state of Virginia in 250 years. John Clayton came back for the unveiling of the new Flora of Virginia with his flora from 1761. But floras are problematic. They are heavy, they're big, they're cumbersome. As I said, they get old. Flora North America is a wonderful project, but not certainly practical to use as a field tool with 30 volumes. And a penstemon key that involves 238 species of penstemon. One would never really want to have to go through that key. What we really want is Spock's tricorder, but Nobody has really invented it yet. We want to be able to point it at a planet and know where all the biodiversity is and how many species there are and how many of them are intelligent. But we can move in that goal, towards that goal and in that direction. And so what we've been trying to do is to develop Floras, apps, and guides, sort of all kinds of products based on the same system. And that system is Flora Manager. It's basically an access database in which we manage all the information from which we produce these tools. And this is what some of the data structure looks like. A sort of simplified version looks like this. So we write keys, we produce maps, we make taxonomic changes, we associate various pieces of information, traits, and so forth with species. We rearrange families in general when new publications come out. This is what the entry screen, one of the initial entry screens, looks like for this. So you click on any one of these buttons and you get additional entry screens. This is the screen, the species detail screen for Magnolia macrophylla. So there's text in here. There's synonymy against a long list of other floras. You can click to the traits button and see traits or populate traits. The keys are accessible from this screen as well as in other ways. Mapping is done on this map screen. It's done by pointing and clicking and all. And the key looks like this. We can remove key leads or add key leads easily with these little buttons and all. And so this is how we have created the 25 floras, 27 floras covering different parts of the Southeast at different scales, from the full regional flora to sub-regional floras to individual state floras. And this slide is out of date. We've now had over 7,000 downloads of the PDFs from the site. Those downloads have been mostly from Eastern North America, but also from lots of other places as well. And this is a heat map, also out of date, of where floras have been downloaded from. So these are all available, as Bob mentioned, from the North Carolina Botanical Garden website, and they are free, open access. So they contain, increasingly, we're adding in identification notes. There's a dichotomous key, there's information on the species, there are range maps that cover the distribution in the Southeast and also show where outside of the Southeast the species is distributed. So Huperzia lucidula also occurs in Asia and also occurs in Western North America, in addition to northwards and northwestwards in Eastern North America. And we are at work on a variety of derivative and collaborative projects. The Flora of Virginia was really one of the first of those. We're now working on a Flora of Delaware. Bill McAvoy from the Delaware Natural Heritage Program, Amy Highland from the Mount Cuba Center, and I are developing a Flora of Delaware that will be a print flora as well as released in digital forms. A guide to the Flora of Arkansas. Arkansas has never really had a flora. A Flora of Georgia, Max Medley and I are working on. The flora has been used as the legal basis for Pennsylvania's state listing of rare, threatened, and endangered species as a reference for its the taxonomy. And we've also been working and developing florulettes or florulas of individual park units for the national park system, so that they can have those for the public and for their resource managers, an actual customized flora for the 573 species in a particular national park unit like Little River Canyon National Preserve. Because of the way we're managing the floras as the flora information as a database, we can customize the output in a whole variety of ways. And recently we have started adding in pictures to the customized output, which clearly makes the floras more user-friendly for the less botanically trained individual. And the keys, when we produce these custom products, the keys basically automatically subset to only the species that need to be in the key. So if you produce a Flora of Texas, the key subsets to only the 3 species that are present in Texas. If you produce a Flora of the DC, Maryland, and Virginia areas, the key subsets to the 6 taxa necessary for that area, whereas the full Magnolia key in the Flora Manager or for the full Southeastern Flora would be 12 or 13 species. Okay, so apps. We've talked about floras, we've talked about PDFs of floras. Let's talk about apps. So I hope some of you have had the opportunity to use the Flora of Virginia app. It includes a number of innovative things. It includes all of the information that's in the big printed Flora, except you can carry it around on your phone. It is resident on your phone and therefore does not require internet access. And it includes all the dichotomous keys. It has many more illustrations than the printed Flora has, and all the rest of the content as well. In addition to the dichotomous keys, it also has graphic key or multiple access key. And the graphic key operates by starting off with the 3,164 species in the Flora of Virginia. You pick a plant group like ferns or lycophytes or woody plants, woody broadleaf plants. So here we've selected broadleaved woody plants. And just by clicking that one button, you've reduced from 3,164 species to 474. As you go ahead and click other things, other pieces of information about the plant that you have in your hand or that you're standing in front of, basically it keeps on subsetting the flora and eliminating possibilities. And so once you have chosen a broadleaved woody plant with opposite leaves, with simple, palmate, lobed, and toothed leaves, you're down to 10 possibilities. You can then click show, and it'll show you the 10 possibilities all in the genera Acer and Viburnum. You can then click on thumbnails and identify it. A new thing that we're going to be adding into the apps that I'll be talking about in a minute is we will be adding in, actually at this point, rather than just going to thumbnails, you can also click on a button which will produce a key on the fly to the remaining species that you have. So if you have 10 species left, it'll produce a key to those 10 species. Once you think you have an ID of Acer nigrum, you can look at pictures, you can read descriptive information, you can see the sort of breadcrumbs of how you got there through the graphic key. You can see the distribution map in Virginia, and therefore check your ID. So we're going to be developing apps similar to that across the full Southeastern region, all or parts of 25 states at all. So not content with floras and not content with apps, we also embarked on writing a wildflower guide. And you all in New England are probably familiar with a parallel guide that was done by the New England Wildflower Society et al. So here are the authors of our sort of mid-Atlantic guide. And you might say, why a new wildflower guide? Well, I grew up on wildflower guides, and I feel like they're a gateway drug to serious botany and to serious natural history in general. Peterson wildflower guides, was the only thing available back when I was a kid, and they had a lot of limitations and weren't nearly as good as a lot of guides that are available now. But there's a large set of users who we want to know about natural history who are never going to pick up a 1,200-page tome. There are some who are never going to open an app. I think our philosophy is to put the hay where the horses can get it, and horses with different tastes and different approaches to technology and paper and level of detail. So the goal of a new wildflower guide is to cure plant blindness, provide a basis for citizen science, engage young people with natural history, develop a constituency for land conservation, and so forth. Luring people deeper into caring about the real world. So this was part of a series done by Timber Press and covered from New Jersey to Georgia, and has a fairly simple kind of multi-access key-like approach, sort of similar to Newcomb's Wildflower Guide. Those of you who have used that, which is probably very many of you, where rather than just having a big section of white flowers that you flip through, you are asked to answer a simple set of questions like flower color, flower symmetry, number of petals, leaf type, leaf arrangement, and leaf margin, that basically narrows it down to 3 or 4 or 10 or 1 possibility on a couple of pages in the guide that you can then compare it to. So, what we hope is a taxonomically sophisticated, ID sophisticated, but easy to use wildflower guide. So for hepatica, blue flowers, flowers radial with 7 to many petals, leaves deeply divided, basal segments entire, takes you to page 435 with just a few species to look at. So I want to close with sort of coming attractions and talk about what we're up to, where we want to go with all this. And that is to develop Flora tools for all levels, including the professional. And I think in the year 2022, it's not too much to expect that floras are readily updatable. They can be kept current and reflect changes in our understanding of taxonomy, changes in what species have naturalized in the flora, and so forth. So information that changes can be revised. We can provide current taxonomy, and we can provide a crosswalk to alternative taxonomic systems. You don't like Weakley's taxonomy and want to use Cronquist's 1991? Okay, well, you can do that. We want to provide information that is broadly useful to the professional. One of the key things that, so to speak, that I have always tried to do in the Flora of the Southeast project is to write keys that are based as much as possible on vegetative features. You shouldn't have to, at the beginning of the key, answer some technical feature about the ovules, how many ovules there are, that is going to require cross sections and a microscope and so forth to tell this family from that family. You should be able to key a plant based on the obvious features that you see in front of you, its habit, the leaf arrangement, etc. Now, you can't always do that, and particularly when you get into grasses and other technical groups. There's no dumbing down of the fact that you will need to look at detailed features and so forth, but those can be pushed later into the key so that your process gets you down to a relatively small number of possibilities before you have to go in deep into details. And then at that point, perhaps the possibilities are also limited by geography or habitat or other features like that. So providing traits, G ranks and S ranks, wetland status, coefficients of conservatism, FQI, heliophily or grasslandiness ratings of species. All of these are things that are needed for the conservation professional for land management, as well as of interest to the casual user. And Floras have not tended to have this kind of information. You all in New England are a little bit spoiled because of the Go Botany website and Flora Novae Anglii, probably have provided this better than some aspects of this better than in most other places in the country, with, again, probably the exception of California, with the Jepson Flora Online and website resources provided by UCal Berkeley and the California Native Plant Society. But most of the rest of the country is poorly served with availability of these kinds of information. So we are going to be developing additional plant identification apps using graphic keys, but also with alternative systems for ID. There will be a North Carolina Botanical Garden enhanced website for the native and naturalized flora of the region with 50,000 plus photos available. The new Southeastern Flora will continue to produce in PDFs and to develop collaborative projects like the Flora of Delaware. And in order to do this, I've put together what I really think of as the dream team, Floras apps, to develop Floras apps and website for the full region and state by state. We're working with Katie Gibson of High Country Apps in Bozeman, Montana as the app developer. Michael Lee, who developed the Flora Manager system, is now full-time employed by me. And then we have a range of great botanists, including, as Bob already mentioned, several with New England roots, Bruce Laurie and Richard LeBlanc, Scott Wards from New York. I've got a grad student, Aquila Ledesma, a computer scientist working on artificial intelligence and machine learning to see what we can do to make apps like iNaturalist smarter about their ability to ID plants, and all supported, of course, by the herbarium and so forth. So we got big plans. So the apps will probably have 3 ID methods: dichotomous keys, graphic or polyclave keys, and artificial intelligence. We hope to interlink the apps with iNaturalist so that you can make iNat observations directly through the app. There will be multiple photos per species, diagnostic photos showing diagnostic features. All of these resources will have traditional information like habitats, maps of distribution, native, non-native status, taxonomic discussion. We'll have a clickable illustrated glossary. About 7,000 references are cited in the overall flora, so those will also show up, and we'll have the detailed crosswalk to other floras in the region and to taxonomic literature, monographs, and so forth. So all this is being made through kind of a diverse revenue stream. I don't want to get too much to the money, but it's worth mentioning. So we're working with a lot of collaborators, contracts with National Park Service, funding from the Mount Cuba Center and the Flora Virginia Foundation, as well as the Southeastern Grasslands Initiative. We've had contracts from various state governments in the region because they recognize the need for customized floras for their state and so forth. And we've had very generous funding from a particular individual private conservation philanthropist who sees the value of this as for leveraging conservation, which is his real passion. And we're looking to develop additional collaborations across the region to put in the hands of people who need it what they need. So I'll close with this slide, this great quote that I'm sure many of you all have heard before by Baba Dioum, a Senegalese forester in the 1960s. In the end, we will conserve only what we love love only what we understand, and we will understand only what we are taught. And to me, developing these better botanical information resources in a whole variety of formats is my way to try to do that. thanks, and I'll be happy to answer questions.
Very exciting talk to a lot of us who work in the field a lot, and the idea of all those apps is just really thrilling, and the more mathematical approach to keys, some of which I've used out of Louisiana State for sedges, where you can click on various measurements and it clicks right down to the few species. So we're all excited about that. I can see from the chat room that we're very excited. I want to start with my own though, and this is related to everything you've said, but it's not directly anything you've discussed. But I really want to hear your approach to this, or your sense of. And one of the things that we have been hearing a lot, especially from the Nature Conservancy, about preservation of biodiversity, is this concept of resiliency, especially landscape resiliency. And many of us here, well, I shouldn't say many, several of us here in New England are very concerned about like our hotspots of diversity, Martha's Vineyard, Nantucket, Cape Cod, that are relatively flat. And these do not fit into the concept of landscape resiliency. In fact, they're threatened directly by sea level rise. So, and I know, like, I think in your area, I think of like the Carolina Bays. I mean, they're very flat, obviously, must be highly diverse. Where do you come down on that, and what do you think about that?
Yeah, that's a good and a tough question, and one I think all of us wrestle with a bit. I guess I would urge us to be cautious about thinking that we know more than we really know about the future. I think these analyses and projections, there are a lot of assumptions. So if something is at 1 foot above sea level, maybe we can legitimately and with some certainty say, yeah, that's going to be a problem. But I've heard projections. I've heard suggestions, for instance, that the high elevation flora of the southern Appalachians, things like Geum radiatum, and all the endemics that occur on these high elevation rock outcrops, or even Abies fraseri and so forth, that we should just kiss all those goodbye and forget about any effort to conserve those species. Well, they've been there adapting to different climatic regimes for tens of thousands of years already, and we don't know how they're going to respond, how these systems are going to respond, or how the component species are going to respond to climate change and so forth. And I guess to get a little more philosophical, I guess the way I tend to think about it is I'm just going to do the damnedest to pass on as much biodiversity to the next generation as I can. And I hope that they will then do the same, and the generation after that will do the same, and so forth. And desperate measures at some point may be needed. And you can get into all sorts of complicated discussions about assisted migration and so forth, which I also have deeply mixed feelings about. But I think we need to be humble about our ability to predict the future. One last comment about that. So some of the greatest biodiversity hotspots in the Southeast are in the coastal plain and at low elevations in flat landscapes. The Apalachicola National Forest in the Panhandle of Florida, the Green Swamp area in the coastal plain of North Carolina, et cetera. Thousands, literally thousands of endemic species associated with the coastal plain. Plant species alone, thousands. Some of us who are gray-haired or don't have any hair left are used to seeing mutual fund prospectuses that say, what's the line? Past results do not guarantee
Future success.
Yeah, future success or whatever. And except that I think maybe in biodiversity, that might actually be really true, that past results do suggest something. So these areas, they were resilient in the past, they were resilient through past changes. And why shouldn't we also have the expectation that they may be more resilient than we expect through future change? So let's be humble about over predicting and writing things off. Yeah, that's enough, I guess.
Yeah, that was really a great answer. I'm glad I asked. A question from our new president, Matt Charpentier. He asks, Alan, do you have suggestions for species which have recently been recognized, many of which you mentioned in a slide, which we should keep an eye out here for in New England?
Oh, well, yeah, that gets into a lot of specifics, but of course, I would say that particularly in some of the groups that are pretty technical, things like ringcosporas. When new ringcosporas get described in the southeast, I would look for them in New Jersey or out on in pond shores at Cape Cod and so forth. Yeah, I'll take that offline with people to talk about some specifics, but certainly, yeah. I think not only are we discovering new species, but we're also discovering new components of the distribution of species that we didn't recognize before.
Yeah, I think that's true. I'm always suggesting new species botanists to look for here, things like Floerkia, which we don't have in Massachusetts yet, and Pyxidanthera is only in New Jersey and a little bit in Long Island, but should be in our, especially now that we have more fire on the landscape. There's a few others and one, in fact, that Matt suggested that I can't remember the name of.
When I was the botanist for the North Carolina Natural Heritage Program, I used to maintain a hit list of species to look for, about 50 things that I thought could turn up in North Carolina, and a fair number of them did. And that is part of you don't see what you don't believe you are going to see. So it helps to be aware of possibilities. So. Yeah,
for sure. A couple questions from Lisa Stanley. Is there a good app for the Smokies yet?
No. So certainly the apps that we're developing, the one for North Carolina should probably be out in about a year or a year and a half. So that'll be available, and that will cover the Smokies well. You'll basically be able to zero down to the mountains of North Carolina or the mountains of Tennessee for coverage of the app. You can subset the app via geography very easily. So
are these going to run about the same price as the Virginia one?
We haven't fully decided on the pricing. I think we're going to have a slightly complicated pricing structure to not penalize people who want a large area, but also not penalize people who they're only going to use it in New Jersey, so they only want the New Jersey app. So I think a state standalone is probably going to be less than the $19 of the Flora Virginia app, and the full region may be more like that. So it may be that you get the umbrella and then you have add-ons for a few bucks each.
Mm-hmm.
We haven't quite decided on the pricing structure yet.
Another question from Lisa, and you touched on this a bit, but what are your overall thoughts on iNaturalist? Obviously, and we mentioned it in the very beginning too. From my point of view, it seems like it has a lot of potential, but there's also a lot of disarray, you might say, in that I participated a little bit in that ideathon and ended up with, just a few people who had only one image per plant, and it was all out of focus. And it's just like you want to tear your hair out sometimes. But I know you have quite a few observations, over 4,000.
Yeah, I have sort of Mostly a love relationship with iNaturalist, but a little bit of a hate relationship too. I think it's complex. When they first introduced their AI, I thought, boy, this is going to work for mallards and mockingbirds, but no way is it going to work for plants. And I'm actually overall surprised at how well it works for plants and all. And you know, I think one has to have maybe modest expectations about the level of taxonomic accuracy that you're going to get, particularly in technical groups like sedges and grasses and so forth. But on the other hand, I think as a tool to get people interested in natural history, I think it's great. There's, you know, there are generations, including my own son and daughter, who probably will never pick up a botany field guide or flora, but who might use an app and all. You know, I have found that it's actually of considerable scientific use. I and an undergraduate student were looking at Hydrophyllum virginianum and two varieties that differ in flower color. And on herbarium specimens, the flower color fades and isn't very definitely visible and so forth. But, you know, we could scan 4,000 occurrences uploaded in iNaturalist of that across a broad geographic range and see that the flower color difference was, you know, geographically very strongly correlated that we never would have been able to do with herbarium specimens. So buyer beware, lots of good things about it and also some bad things.
Yeah.
I don't know if that answered your question, Lisa.
Yes, I mean, here's another approach to that question. Would you encourage more of our members to use it?
Yes. Yeah.
Great. That sounds good. I mean, that's a definite yes or no. So I like that. Professor Bellamar wants to know, given the substantial number of narrow endemics in the Southeast and threats from habitat loss, climate change, et cetera, are there concerted efforts yet to begin conserving some endemics ex situ at the University of North Carolina Botanical Gardens, seed bank, et cetera? I think the answer to that is yes.
Yeah, the North Carolina Botanical Garden is a CPC, Center of Plant Conservation, actually founding member. We have a lot of seed banking of rare species as well as a broader set of native species for restoration use and so forth. But yeah, we're definitely doing that. We have our hit list of species. As the Fish and Wildlife Service listing process has sort of gotten more and more politicized and gone further and further awry, you know, we don't wait for federal status. We really look more at all the G1 and G2 species with 3 or 5 or 15 total populations as high priorities for in situ conservation as the primary conservation means, but also ex situ conservation as a backstop to be sure that we have a seed source remaining for those species if they were to go extinct in the wild.
Great. I'm going to have a couple more and then we'll start to close it up. But a couple more great questions here from Matt Sharp and TA again. Do you have suggestions how we here in the Northeast can efficiently keep up with taxonomic revisions occurring in the Southeast, aside from the literature review at the species level or waiting for a new regional flora to come out?
Yeah, so in the next week or two, I'm going to issue the 2022 interim edition of the Southeastern Flora. And I am planning on including in that actually a summary of changes. And I think going forward, that will be a plan, is to always have that summary of changes. The Weakley's Flora of the Southeast Facebook page, when there are new species described, I generally post them on that. It's a difficult thing to sort of fully summarize some people want to know, oh, the key changed, or other people just want to know, oh, a species changed. Some people want to know this new non-native species we've decided to include in the flora for the first time. All the different kinds of change. It's hard to decide sort of for different users what everyone wants to see, but we're going to start producing some summaries of those kind of changes, which I think may be helpful, Matt, to the sort of thing. I've definitely heard from a lot of people that they wish that they had an easier source for things like that. So hay-scented fern, Cytobolium punctolobulum. Are you ready for that? Dinstidia is a tropical genus that doesn't have much to do with our temperate thing, which is related to a couple of species over in East Asia. Another one that's going to be a shocker for some people. Do I want to bring this out at the end of the talk? I guess you all can't hit me with rotten tomatoes through virtual means, right? But New York fern, Thelipterus, Parathelipterus, some recent monographic work on the Thelipteridaceae by Susan Fawcett and Alan Smith now place that in Amauropelta noveboracensis. Also related to some Asian species, as well as some actually some tropical species as well.
That one I think you can at least pronounce, right? Amauropelta.
Yeah.
One from Matt Arsenault. In general, do rare plants see much protection at the state level in southeastern states? And does that include things as part of environmental and land use development permit process?
Well, it's complex. I'd say yes, it's somewhat variable from state to state, but there is a good deal of attention. In North Carolina, we have the Natural Heritage Program and also a plant conservation program that handles the official state listing. We have, I think, over 500 species listed as endangered or threatened in the state, plants listed as endangered or threatened. As is usually the case with plants, the amount of protection that generates is limited. It's mainly trade and on federal or state lands. It doesn't affect private property rights, et cetera. There are a number of states, including North Carolina and Alabama, that have pretty robust land acquisition programs. In North Carolina, I served on the Natural Heritage Trust Fund board when we gave away, over a period of 6 years, about $500 million worth of funds for state land acquisition of natural areas, additional conservation areas. So there is a lot of active conservation work and a lot of additional land being put into protected status in the Southeast.
Yeah. Great. I think one final question, which I think could be succinct, and that is from Joyce Reddick. She says, where does consideration of pollinators fit into the modern species concept?
So species biology, and particularly aspects of it that have to do with reproduction, can be absolutely critical determiners of crossing, and therefore whether something is isolated or not isolated. So pollination biology is a really important factor. It's also a very important factor for applied conservation. So I have a grad student and he's also a colleague at the North Carolina Botanical Garden, Mike Kuntz, who's working on a species of astragalus that grows in sandhills, and the populations are very small. They set seed poorly, so he's been doing detailed study of the pollination ecology of the species to sort of understand what's going on there. We were also involved in another recent study of Venus flytrap, which sort of interestingly maybe not surprisingly, but interestingly discovered that the insects that it traps are not the insects that pollinate it. There's very little overlap. So it's mainly catching ants and spiders that are crawling along the ground, and then the flowers that are a foot higher are attracting sweat bees and beetles and so forth that are almost never show up in the traps. So Venus flytrap is not eating its own pollinators.
Yeah, you would hope. Yeah. Oh, well, this was really great, and people are commenting online that thanking you profusely for coming to speak to us today.